All right, good morning, everyone. So before we get started, let's see. Before we actually get started, as you guys know, we will be going in person as of next week. So we will switch into that modality. We will also be soon transitioning to a few lectures that are going to be seminar style. So your next assignment will be focused on some articles that I will have you guys read that will be the focus of a discussion. So I will have either that assignment ready for you for tomorrow so that you'll have enough time to be able to read the material. And pretty soon I will be administering a sign-up sheet for the set of seminar topics that you guys will be participating in. So in general, every person will be participating in a presentation of a article under a specific topic. So you will see the different topics that are offered. You can look at the syllabus and see the different general topic listings. Everyone will be working in a pair. So every presentation that you give will be with a partner, and so I will explain to you guys the guidelines for the preparation of these seminar presentations, and there may be a situation where you might have to pair up with me. Okay, so if you have that lack of fortune to have to be paired with me, well, we will figure this out. Anyways, that is coming up so that you guys are aware about these two things that we're going to be working on. All right, so let's go ahead and get started. And what we're going to do is continue on from our discussion last time where we were talking about general transport of solutes. And as we're transitioning into discussion about transport through membranes that have to be, that involve metals. And so at the end of our discussion, we were talking about passive transport versus active transport. And one of the major distinguishing features is that when you're referring to a passive transport, you're looking at something that is going to be thermodynamically favorable. And this will lead to a movement of a solute so that you reach an equilibrium in terms of the concentration. You don't actually go above that. Whereas when you're focusing on a process that is actually active, well, then this will be a movement of a solute that requires some form of energy, given that the actual movement of that solute is not thermodynamically favorable. So you couple this with a chemical reaction that facilitates the energy needed to move the solute, or you couple this with the movement of another solute that is thermodynamically favorable. And so these are just the two major types where you either couple this to the breakdown of ATP, and so that hydrolysis process leads to release of energy that can be taken advantage of, or you can move along with the movement of a given species that is falling down its electrochemical gradient. So when we're talking about this idea of the transport of a polar or charged solute, okay, so here we're moving into this idea of a facilitated process. So there are membrane proteins that will facilitate the diffusion of polar charged solutes. These are called transporters, or if you want to think of them as a type of enzyme, they're called permeases. So they're enzymes because they will speed up the process of the movement of that given solute, the given species. But they're not enzymes in the traditional sense that we know as a conversion of a substrate into a product. And so in this context, those permeases will move the solids in the direction of or against the electrochemical gradient. And so what we're looking at the right is a depiction of this process that is being accelerated. So what we're seeing is how something that is polar or charged. And so here we can start to think about metals, metal complexes. Typically, when we're talking about a metal ion, it is positively charged. Metal complex, that can vary in terms of charge. And so now we have to think about, well, how is this being facilitated? So in the absence of a transporter, then what we're looking at is there are going to be some amount of energy penalty associated with this movement. And so if we look at the schematic or the representation at the top, here we're looking at a polar species that is hydrated, okay? And so in order for that particular solute to be transported, there has to be a loss of that hydration, given that that solute is going to go into the lipid bilayer. And then when it crosses through, then you get the rehydration process. And so you have an energy penalty there associated with, and if you look at this reaction coordinate as a representation, you have this increase in energy, this activation barrier, that has to do with that loss of hydration in order to then go through the nonpolar environment of the lipid bilayer. And so that's the energy penalty that you have to overcome. But then if you look at the representation at the bottom, the role that the transporter or the permease is playing, where essentially what's going on here is that now you have a protein that is going to facilitate this movement of that given polar species. So yes, there is probably some amount of hydration loss, and so at least from this perspective, but that given species now is going to be introduced into an environment that probably is more favorable, given, let's say, the type of amino acids that are lining that pore. And so now you don't have that high of an energy barrier because you're introducing a polar solute, charged solute, into an environment that is more favorable to it. And so you have effectively reduced that energy barrier, okay? And so because you have effectively reduced that energy barrier, what is going to happen? Well, you're essentially going to speed up the process. You're increasing or you're decreasing the value of K, the rate constant. And so you move through, and as depicted in this reaction coordinate, you're going to end off at an energy level that's equal to the start, because essentially you're not actually transforming the solute, you're simply facilitating its movement from one side to the other through that given membrane. All right, and so in this context then, your delta G double dagger, which corresponds to the activation barrier, will be lower for the transporter than it would be if you were just representing this as a simple diffusion. So again, thinking about metal complexes, here we have to think about the idea of them being charged, and even then, if they're not necessarily charged, you still have an environment that should facilitate their movement. Now, when we're talking about these type of transporters in a very general context, you can look at them from the context of whether it's a transporter for a movement of a single thing, or if the transporter that's coupled with the movement of another solute. And so we have the uniport. In contrast, we have what are known as a cotransport, where you can break them down into the category of a symport, and that is you have a movement of two solutes in the same direction through the actual transporter, or an antiport, where obviously it will be the opposite, where you have your solute going in the opposite direction of another solute. All right. So let's actually clear that up, cancel that. So then the directionality can be with or against the electrochemical gradient. All right, so it's either something that is going to be thermodynamically favorable or not. And so in that context then is how this is being facilitated in both passive and active transport. All right, so this being a transition into discussion of an active transport. Well, in primary active transport, the energy released by ATP hydrolysis, and so here is just a representation of ATP, and you're basically getting the lysis of one of the phosphates, the terminal phosphates of the molecule, leading to the formation of ADP, an inorganic phosphate. Well, that process actually releases a significant amount of energy, so we have negative 30.5 kilojoules per mole. And so that particular energy is what allows for then a solute to be able to move through the transporter against the electrochemical gradient, right? So we see that we have, at least in this representation, the solute at a low concentration moving into a space where it is a much higher concentration. And so this process of actually moving through the transporter, not thermodynamically favorable, but this becomes a possible because of that energy being supplemented to it. Whereas in a related process, which is the secondary active transport, if we're considering the movement of a solute, well then here we have this being coupled to something else. So a gradient of ion S1 has been established by primary active transport. So let's say that we're moving solute 1 according to this picture, okay, and it's moving in this direction. So let's say it's going, let's go the other way. Okay, so we have that moving in this direction, and that's being facilitated by the hydrolysis of ATP. Excuse me. Well, then movement of the S1 will be down its electrochemical gradient, now provides the energy to drive the co-transport of a second solute. All right, so what does that mean? Well, imagine us moving in this direction. So S1 going in this direction from high concentration to low concentration. Well, then that is going to supply energy that's going to facilitate S2 to move from low concentration to high concentration. So that S2 is actually going against its electrochemical gradient. So that is a type of transport that's being facilitated, and is referred to as a secondary active transport. So then when we think about transporters, now trying to look at them in terms of specific categories, distinguishing between channels and carriers. So then here you can break down carriers into this idea of the directionality of what's being moved and what type of facilitation you're providing, whether it's a primary active or secondary active transport. All right, so before we go into those two categories, let's look at a little bit about the idea of transporters, permeases, as I had mentioned previously, facilitating an increase in the speed at which something's actually being transported. And so if you were to follow the kinetics of the rate by which you're facilitating movement, and you were to look at it as it depends on the concentration of the given thing being moved, you would see that this would actually fit a model like the Michaelis-Menten equation. So if you think back to biochemistry, you explore the Michaelis-Menten equation in terms of enzyme catalysis. And so this is the idea of looking at the speeding up of a process, and this idea of possibly saturating an enzyme. And so you're going to reach sort of a maximum velocity of this movement, of this process being catalyzed. And so we see something very comparable in terms of the transporter kinetics, that you can fit it to a Michaelis-Menten equation. So let's look at what we have here depicted in the picture. And so in this picture, what we're looking at is zinc, okay? And zinc is actually being transported by a particular yeast. And so it is a particular transporter that's facilitating this movement. And so it is a ZRT2 transporter. And so what we're looking at is what happens when you're introducing this particular transporter in a given host. So we have an overexpression of that particular transporter, and one in which we don't have that overexpressed. So you have more of transporter versus the concentration of the zinc. We see that we have, as you increase the concentration of zinc, okay, we see that we are increasing in the velocity or the rate of this. But then you're going to reach a concentration range where you essentially are going to basically plateau, okay? And so plateau means that you have reached a maximum velocity. That's represented by this term Vmax. All right, so according to then the fitting of this data, we basically are fitting the Michaelis-Menten equation. Right, so we're looking at velocity as it depends on, in this context, the solute that's being moved. And so there are a few different parameters that we're able to fit. For instance, you're able to fit this parameter here, which basically is the concentration of the solute at which you are facilitating half the maximum velocity. So basically, the way we could be able to see this is that if you have a maximum velocity of 16, this is picomoles of zinc, if we're looking at the units per minute in these given cells, then we want to determine, well, at what concentration does that happen? So we have to see, if you look at the plot, where you reach the half maximum velocity, which would be 8, and then you bring it down here is where you're able to determine this particular concentration at which you're able to achieve the half maximum velocity. So that's that parameter KT in terms of the Michaelis-Menten equation. All right, so what does this mean up here, this part of the plot? Well, that means that you are saturating the transporter. So you're going to reach a point that you simply just can't transport any faster because you have maximized the available I guess space for movement of the actual solute. All right, so now going into this the categorization of the transporters into the channels and the carriers. Well, channels are proteins that form pores in the membrane for diffusion. And so here we have a representation of possible pores that can exist. And so pores open in response to signals. So this can be a membrane electrical potential change, or a signal could be that there is a receptor for a given ligand, and when that ligand binds to the receptor, that then sends a signal for the pore to open up and then facilitate movement. And so we have a few representations here. So like for instance, you have the one that's voltage gated. Okay, so that then is regulated by a change of potential, and so that would lead to an opening of the pore in order to facilitate movement. Or as I just mentioned, the ligand binding, where you have a ligand binds, and then that leads to an opening of the pore, which will facilitate the channel to open up, and then there can be a movement of the solute for a given amount of time until it closes back up. And then there could be other types of indicators or stresses that can then lead to a signal for movement. The thing about channels is that they have low stereospecificity. Okay, some signal has to be reached in order for them to be opened up, and because of the nature of how it facilitates movement, they catalyze transfers at rates several orders of magnitude greater than carriers. So carriers is a comparison of the type of transporters. So they actually approach diffusion limits. All right, so K can be pretty high, can be as high as 1 × 10 per molar per second. So essentially, in terms of how you're looking at this, they essentially would be not saturable. What I mean by that is that you have an opening, and so your solute can go through until that opening closes, right? So you're not really you're not limited to the actual solute and its interaction with the transporter in terms of movement of that solute. But in contrast, something like a carrier, there will be a limit. And so here we have a representation of a movement of glucose, okay, in terms of its interaction with a given transporter. So carriers are proteins with high substrate stereospecificity. So they have a certain recognition, and in this case, that's very stereoselective. So they bind substrates at one side of a membrane, undergo a conformational change, and then release the substrate on the other side of the membrane. So what we're depicting here is a glucose transporter recognizing D-glucose, okay, so that form of the glucose. And so it gets recognized. That recognition process leads to a signaling for a conformational change, and that conformational change will facilitate an opening for the glucose to come through. Then the glucose comes through, and then you have a closing of the transporter itself. All right, so there you see the restrictions that are imposed by the actual solute coming in contact with that protein, and so you can see how that can become saturable. So as a consequence of this limitation, they catalyze transport at rates below the free diffusion limits. So they are much more representative of what we think of a classical enzyme, in that you will observe that saturable-like enzyme behavior. All right, so when we're thinking about types of transporters, you also have what are known as ionophores. So as the name implies, it is something that will facilitate the movement of a charged species, of an ion. And so an example, okay, this could be a diverse class of organic molecules that increase the permeability of membranes to particular ions. So this can be peptide or non-peptide based. So in this category of transporter, there are certain metal ligands that basically play the role of an ionophore. And what I mean by that is that their function is to bind a metal, and then that complexation simply is used to bring in the metal, and then there's no actual active role other than transport that the ligand provides. All right, in terms of where we could see this, the ionophores, well, bacteria are known to produce peptidic antibiotics. Well, they're not necessarily, you know, we have to kind of fine-tune that, but anyways, these are peptide-based ionophores, and so the function of this is to try to inhibit the growth of other organisms, so other bacteria, by shuttling and depleting the levels of important ions. So bacteria are very aggressive about a certain essential metals like iron, to an extent copper, and so they have their own methods to bring in a certain metal that they want, but also to try to limit the accessibility of that metal to other bacteria or even hosts. And so an example of this is the sinomycin complex here. So that's basically this peptide that can recognize potassium. And so essentially what happens is you have a movement of this ionophore to the membrane, okay, that then will bind to the potassium, and that's going to shuttle it in, and then it will actually bring in the potassium, and a net effect of that is you have a decrease in the amount of potassium that's accessible in that extracellular space. Another type of transport that's possible is a receptor-mediated endocytosis. And so here you have the actual membrane, cell membrane, playing a very vital role in terms of facilitating movement. So in the process that I already described, the membrane always plays an important role. But here, the membrane is even more active in terms of this process. So in general, endocytosis is the process by which cells absorb molecules by engulfing them. And so they use the plasma membrane. And this process is coupled to a ligand binding to a receptor, and this ligand receptor, so ligand in this context is not like a metal binding molecule, but rather it's something that is recognized by a receptor. This interaction between ligand and receptor triggers delivery of the ligand into the cell. So in this representation here, we have a receptor represented in green. It will recognize a particular ligand in this context. And then that is going to lead to a process in which the cell membrane will engulf that protein substrate complex and will bring it into the intracellular environment. So this can fall into two categories, one which is clathrin dependent and one which is clathrin independent. So the clathrin dependent process is one that involves trying to preserve the receptor that is actually facilitating the movement of the substrate. So clathrin is a large protein that forms a coated pit on the inner surface of the plasma membrane. The pit fuses to the membrane and forms a coated vesicle. So this essentially is a depiction of the vesicle. And you have the red little indicators here that are supposed to represent clathrin as a protein that's coating the outside of here. And so the reason, at least in this process, these are present because they serve as indicators. An indicator that the contents of the vesicle are to be protected. They're not to be degraded. Because if they're not there, what ends up happening is that there is going to be an enzymatic process to degrade the endosome, and basically will open them up. But here, rather than degrading the endosome, there's going to be a mechanism that's going to lead to the release of the solute into the cytosol. And so you have the clathrin independent processes where essentially there's going to be a degradation of the endosome in order to facilitate release. All right, so now with this context in mind, then let's talk about mobilization of metals. Okay, so mobilization, transport. So this sounds very similar, and to an extent they are similar, but there are some really important distinctions here. So mobilizing basically is the process by which you make a metal bioavailable, accessible, right? Transport is once that becomes accessible, it's the movement of it for it to be able to reach given targets. And there are three key ways that organisms mobilize metals to make them bioavailable. So here I'm going to give you just the overview. You have chelation. So as the name implies, you transform the metal into a stable but moderately labile formulation. Okay, what does that really mean? Well, stable means that you're going to form a compound with that metal to keep it protected, okay? And moderately labile means that you want to be able to bind it with enough affinity, but you don't want to basically lock it into that complex so that that metal cannot be released and be useful. All right, so stable and moderately labile formulation. Another process is redox, where you either facilitate the reduction or oxidation of a metal, okay, to an appropriate oxidation state for specific transporters. Okay, that's going to be a critical event. And also, this redox process can also facilitate the functionality of the metal. And then another process is acidification, where you solubilize a metal at relatively high levels, and then you prevent formation of hydrolysis products that are typically quite insoluble. Okay, so oftentimes metals are not by themselves in solution at a pH of 7.4. And I mentioned pH 7.4 because that is the pH of blood. They're not very soluble at that pH. And so the precipitation, okay, so the coming out of solution can actually be a way in which the metal is no longer accessible, no longer able to be mobilized. And so acidification is a way in which you can try to take advantage of an insoluble form of a metal, okay, in order to then get access to it and then transport it. And I would say that oftentimes you have a mixture of these methods in order to facilitate the mobilization of a metal. All right, so let's look at this idea of a chelation process. Okay, so again, chelation, redox, and acidification. We're going to focus on chelation. So chelation by siderophores. I'm being specific here. So siderophores are chelators, and they are produced by primarily you see them being produced by bacteria, and they're used by other organisms. So they can be used by bacteria that primarily produce them, fungi, some plants, and marine organisms. The function of these molecules is to be able to acquire iron and facilitate its transport. It turns out that there are hundreds of known siderophores, and people are still trying to explore additional ones, especially as people are trying to understand iron processes within different organisms. And so I'm going to just highlight a few important iron binding moieties. Okay. So the two that are depicted in these molecules, catechol and hydroxamic acid. So in the siderophore on the left, enterobactin, this is a very famous one. Okay, so this particular siderophore has two major features represented here. So you have, has a major moiety, which is the catechol moiety. Okay, so this is the catechol moiety here. And so what that is this characteristic functional group where you have these two adjacent OH moieties present. And so they are very good at metal binding, particularly hard Lewis acids. And so this particular molecule has three of them. Okay, and so each of these catechols can serve as bidentate ligands. If you have three of them, okay, then this particular molecule can serve as a hexadentate ligand. Right? So you can bind all three of those catechols onto a metal, onto, for instance, iron. And if we're talking about the function of siderophores as iron binding. On the right, we have desferoxamine B. Okay, so this particular siderophore features this hydroxamic acid moiety, okay, which when it coordinates would become a hydroxamate. Hydroxamate because you have a deprotonation of the proton. So you see the moiety present throughout the molecule. So you have it there, you have it over here, you have another one here. Okay, so you see how this too has three of those moieties present, and how that can fill is a representation of the coordination of iron at these by these two siderophores at the catechol moiety and also at the hydroxymate moiety. All right, so typically what you're going to see are monomeric structures. And so what I actually mean by that are you're going to have these mononuclear compounds that are their own entity, so monomeric, as opposed to being dimeric, trimeric, polymeric. And so what you would, the typical representation of this in terms of the compound is to use what is known as a metal ligand formulation, which metal ligand hydrogen formulation, which is how many metals are in the compound, how many ligands are involved, and how many hydrogens are associated with the ligand. And so what we have depicted for the iron interrobactin, or the ferroxamine B, is a representation of the coordination of the iron in a 1-1-0 formulation. Okay, so metal ligand H1-1-0. So what does that mean? Well, that means that you have one metal, one ligand, and no protons on the ligand. Okay, where's that proton coming from? Well, let's go back for a moment. If we go back here, let's clear this off, you will notice that the catechol moiety has 2 protons. The hydroxide moiety has a proton. These particular protons dissociate when they bind the metal. Okay, so if those protons were associated with them, we would actually have to count them. So in this context, then we keep track of how many protons are part of the coordination of a given metal. So when we have the representation MLH110, that means that all the protons from the siderophore have been dissociated. Excuse me. And then there's possible stereochemical preference.
Professor,
Yeah, go ahead.
when it's 1-1-0, it's one metal, one ligand, and 0 protons.
That's correct.
Okay.
And then the ligand, because the ligand is hexadentate, then what we're looking at here, at least in these pictures, is a coordination number of 6. So it's a little, if you look here, this is where the iron is located, okay? Then you can see where we have that coordination number of 6, because you have 3 of those molecules surrounding the metal center. Similarly over here, you have the coordination number 6. You have 3 of those hydroxymates coordinated to that iron. Okay, and so depending on the configuration of the ligand, how it's actually coordinated to the metal center. So this is something that I'm not going to necessarily cover, the isomerization that you can get in terms of how the ligands will coordinate the metal center, but that there is going to be isomerization possible. Well, in terms of the recognition of these molecules in their iron form, there may be some amount of stereochemical preference, where in this context the delta configuration observed for iron interrobactin is observed much more than the lambda version of that complexation. So a chiral recognition may be important for iron transport, because we have to think about, well, how are these molecules, and particularly how they're recognized by the protein, how is this even possible, and how can you possibly avoid other metals getting in? So there's some amount of recognition on that level. All right, so another way that we can represent this in terms of the general affinity of these ligands is by looking at their affinity constant, and we're going to actually talk a little bit about how do you tabulate affinity constants. But I'm going to show you this this table here. So here you have what is known as log beta 110. So beta, depending on if you've seen this in your previous courses, is basically an affinity constant K. It's a type of affinity constant. And so beta is more of an accumulated affinity constant. In terms of the context here, it is the affinity constant of the one metal with the one ligand, where the ligand doesn't have any of the protons. So here you can get a measure, a relative measure of that affinity across different siderophores. So interrobactin, deferoxamine B, ferrichrome, arabactin, and so forth, right? And so we get a general sense of how strong that particular molecule is for iron, the stability constant. So we get these values. So you can see that the enterobactin iron complex is extremely, has a very high affinity constant, so quite stable. But sometimes something that you can't quite appreciate with these numbers is just like how much free metal can be facilitated in solution. So there's another very related parameter called a PM. All right, so when you think about PM, well, you guys are more familiar with pH, right? The log of the protons in solution. A very similar concept, which is a measure of the concentration of the metal in the actual solution. Okay, and so here, what we're measuring here is how much relative free metal, okay, our metal available in solution is there really going to be, a free metal. And so the higher that number is, right, the lower the concentration the metal is going to be. So it'll be like something like this. So that tells us another way that we can tell how accessible the metal will be given the ligand that it's actually coordinated to. So you can see that enterobactin, very effective in terms of binding the iron. And then that's going to be very useful for bacteria if the bacteria wants to be able then to grab that iron and take advantage of it. And so here on the right are the moieties that are available in these corresponding siderophores in order to facilitate binding to the iron, and how many are accessible. Whereas in enterobactin, there are 3 catecholates available. In some of these other siderophores, you may not have as many accessible moieties. All right, so key point here is this relationship. The higher the PM value is, the lower the available iron in solution, the lower available free iron in solution. Okay, so it's a good parameter that we can use to gauge. All right, so let's talk a little bit about this idea of then the uptake of iron. So we're talking about metals becoming accessible. So the primary function of the siderophore is to get access to iron. All right, so if we're thinking about bacteria infecting somebody, well, the bacteria wants our iron, right? And so our iron, and we're going to talk about that in a moment, where our iron is even available. They're going to try to scavenge, take away our iron. And so they use the siderophore for that purpose, to mobilize the iron to make it available. But then how does that actually come inside? So we'll very briefly talk about this. So you have these outer membrane receptor proteins that are going to recognize the ferric siderophores. So bacteria have actually evolved high affinity outer membrane receptor proteins for binding iron siderophores. These are substrate-specific carriers that engage in energy-dependent active transport of iron siderophores across the outer membrane. So this is true of gram-negative bacteria, but I don't want to say that only gram-negative bacteria have siderophores. Just giving you a more specific example here. So here I just have a depiction of the presence of the of transporters that are available to be able to recognize, or at least a depiction of the composition of the cell, so that you can see what are the processes that have to be involved in terms of movement of an actual iron ion. Well, here's an example of one transporter. So this is the FHUA that basically will bind the iron complex. So this particular protein is from E. coli. And so this is a structural core, consists of a 22-strand membrane-spanning anti-parallel beta barrel. So these are the type of secondary structure for the protein, as it basically is a transmembrane protein. The able form of the protein has a conformation that basically blocks the periplasmic side of the cell. So we're looking at here. So basically, let's imagine that this is going to be our transporter. This is just a cartoon. So we have the periplasm. So when there is no iron bound, this will be basically blocked. It's not going to allow anything in. However, when you have the ferrochrome iron getting and bound to the actual protein FHUA, what that induces is a conformational change. And that conformational change will then lead to an opening of that protein so that you can actually have movement of the iron.
Professor, there's some static or some interruptions in from your part. At least where I'm receiving.
Then we have a transport of the. Let's see. What about now? Can you hear me?
Ahora sí.
Okay. So what I was mentioning, should I go back a step?
Yes, please.
All right. Should I go back here?
Yeah, around that part, please.
Okay. All right. So what I was discussing was the movement of iron through, in this context, iron siderophore complex, not just the iron itself. And so what I was discussing was what has to happen, at least in transporter that can recognize an iron siderophore. And so what happens is that once that recognition occurs, that's going to lead to an opening. Okay, so that now the complex can actually go through. So now there's an opening and it can actually go through. The next thing that's going to happen, as I was just describing, was that there's a protein that's going to pick up that iron siderophore complex. And then that protein is going to help that iron siderophore to be able to cross into through the inner membrane into the cytosol. So this is where I was just mentioning that this can happen in two different ways. So let's go to that slide. So this can happen in terms of the iron itself being transported through, or the ferric siderophore complex being able to facilitate this transport. So let's all right, so let's show this. So once released to the periplasm space, the complexes are bound by a high affinity periplasmic binding protein. So that's what I was just mentioning to you. So that recognition, that leads to a type of complexation. So here's just at the picture of the iron siderophore binding to this particular protein. This is just one type of transporter. I'm not trying to say that this is true of in general of all processes involving siderophores. That leads to an opening because of a conformational change, a protein conformational change that's now going to facilitate that iron siderophore to move through. At that point, then, that iron siderophore that now is in the periplasm, you have the transport of the iron across the cytoplasmic membrane can be as ionic ferric or ferrous form. That means you have iron either in iron 3 or iron 2 that get transported, or you can have iron siderophore complex, so the ferric siderophore complex that's going to be facilitated through. So these are possibilities of how that iron that's now in the periplasm can go into the cytoplasm. And so typically you have an ATP binding cassette or ABC transporter system that's going to facilitate this movement through. So that just gives us an overview. And so we'll later on discuss a little with more depth this idea of siderophores and other processes to allow metal movement. But it turns out that other organisms can take advantage of these siderophores that are produced by bacteria in order to be able to mobilize iron and transport it and use it. So an example is I'm going to provide in a moment. So we're looking at ferric siderophore receptors in organisms that actually do not produce siderophores. So what does that actually mean? Well, that means that they have evolved their own type of siderophore receptors in order to recognize siderophores. So some organisms will feed off of others because of a lack of their own optimal method for chelating iron. So an example of this is a yeast, so this particular yeast here. And so what it does is that it basically will prey on bacteria and take advantage of their ferric siderophores in order to then exploit the iron that they have. And so this is just a depiction of just this idea of having a siderophore transporter in order to then facilitate the movement of iron so that they're able to use it. Okay, so that just gives us an idea about how you can take advantage by the use of a small molecule in order to mobilize a metal. All right, let's go on to all right, so now that we've talked a little bit about this idea of mobilization, what I'm asking you guys here is to look at this figure, this video, and then what I need you guys to think about is what type of mobilization is being practiced by this fly. So as you guys know, flies will release vomit in order to facilitate, being able to digest their nutrition. So of the types of mobilizations that I listed, what type is being practiced here? Nathaniel says active transport. Remember, transport and mobilization are two different things. So mobilization, what type is being practiced here?
Like acidification?
That's correct. Who was that?
Claudia.
Claudia, very nice.
Wants to solubilize with like the acids from the vomit itself.
Exactly. And so we don't have to do that. Why don't we have to do that, Claudia?
Because we have a stomach that does that for us.
Right, but this is at the level of the mouth.
Because humans, we have enzymes that help to break down, but also we have mechanical digestion, pre-digestion, our teeth. So you got.
You're very formal. Wow. So yes, we have teeth and we have amylase in our saliva. So we have things that allows us to not have to throw up. Imagine we had to do this. Yeah, exactly. So this type of mobilization is acidification. Okay, very nice. All right, so this will then transition into a formal discussion of iron transport. And so when I'm talking about iron, we're looking at either iron in its form of iron 2 or iron 3. Those are the most physiologically relevant forms. I will say that other oxidation states are invoked in terms of different processes, but these are the primary forms that we will, that we tend to work with in terms of mobilization and also transport. So so far, what we've discussed, we've looked at this idea of the basic composition of biological membranes and focused in on solute membrane permeability, focusing then on passive versus active transport, this idea of a thermodynamically favorable process versus one that is not thermodynamically favorable. And then we started to then discuss this idea of metal mobilization. Okay, so we're going to talk about this idea in the context of how we mobilize things. Okay, can you guys give me a second? Because someone's actually waiting for me for something. So I'm going to just pause for one moment. I apologize. Alrighty. So when we're talking about iron, as you guys are well aware, it's an essential metal and it's quite useful for a number of the processes that keep us alive. So here is just a simplified representation of proteins that use iron for a number of different processes. So you guys are probably quite familiar with the hemoglobin and myoglobin, which involve a heme form of iron in order to facilitate iron transport in the context of myoglobin iron storage. You guys probably have also heard of these cytochromes. These are very important in terms of electron transport, ATP synthesis. Okay, so these are things that are really important in the mitochondrion. And so over here, the rest of this are just a number of really important proteins that allow for either the storage of iron or the transport of iron or the use of iron to facilitate other types of molecular mechanisms. All right, so what we're going to talk about is then how do we actually mobilize iron? And so iron comes from the food that we ingest, right? And so here's just a representation of where we can find iron in food supplies. If you guys, so I haven't been able to do so my last two inorganic chemistry courses, but I often will do this iron and cereal demonstration which students can get to do. So I don't know if any of you who have taken my inorganic class were able to do this with me. But basically, you will see that when you read that iron is fortified in cereal, is because iron flakes are actually added into cereal. And so you won't see that with your eye unless you do this homogenization process. You know, remember I was telling you guys about this homogenize a sample in order to extract. Well, you can actually extract the iron flakes from the cereal by using a magnetic bar. So you basically make a mush of the cereal, you take a magnet, and you would just basically pull the iron. All right, so that iron, which is a neutral metallic form, that form is obviously not soluble if you can just simply extract it from water. But what I'm going to mention to you is just the overview of how our body will now start to mobilize the iron that we ingest. Okay, so how do we solubilize, make this iron bioavailable? So you can ingest iron in its elemental form, again, in its neutral form, or as a heme. So heme, a lot of these meats will have the hemoglobin, things like that we can take advantage of, or even non-heme iron in the form of iron 3. Right, so we have the different ways that we can access iron in the foods in the different oxidation states. So then we ingest the iron and it goes into our stomach, and then the process of metabolism, right, is this acidification process. So there we have then what that fly was doing, we're doing that in our stomach, right? So we didn't have to throw it up to do this. And then it's in the stomach, and it's getting acidification process is allowing for the to be able to access iron, let's say in the elemental form, which is not soluble. But let's talk a little bit more then about what was happening here, because you can also have acidification, which will be coupled with a redox event. So for instance, what I mean by that, well, let's say we have the soluble iron from the cereal. Well, in addition to the acidification, you're also going to have a process that's going to oxidize the metal. So you're going from iron 0 to iron 2. Okay, so this is an event that's going to lead to an oxidation of the metal. Well, then what's actually getting reduced? Well, what's getting reduced are the very same protons that are facilitating the acidification process, where you're going from a proton, which is +1, to the hydrogen in the, so hydrogen gas in this context, the hydrogen here is 0, right? So that's actually what's getting reduced while the iron is getting oxidized. So you have this acidification, which is actually coupled with the redox event, right? So these are connected. Then, and so I'm going to emphasize iron 2 is going to be the important form here. Well, let's imagine that we have dietary iron 3. Well, that iron 3 will have to get biotransformed. What do I mean by biotransform? Well, the mechanisms in our bodies have to facilitate its reduction. So we have iron 3. Well, then there has to be some sort of reductase to reduce that iron into its iron 2 form. So as an example, an enzyme, a general enzyme, a ferrireductase, or small molecule reductants in the diet like ascorbic acid, well, essentially to reduce that metal from iron 3 to iron 2. Now, if you ingest something that's iron 2, well, basically you already have the iron in the form that's needed. It's just a matter of now making it accessible in order for it to be transported. So once you have the iron 2, okay, that becomes bound by a protein called gastroferrin, and that will transport the iron to the intestine. So here we're now going to look at the movement through the intestinal cells. You have an iron uptake system that will shuttle the iron from the intestinal lumen into the enterocytes. So enterocytes are intestinal cells, and these are lining the small intestine. And this process is being facilitated by a transporter, the divalent metal transporter, abbreviated as DMT1. So as the name implies, divalent, it means that it's going to have a charge of 2+. So that's why it's very important that that iron is iron 2+, okay, divalent metal transporter. So as I mentioned, this is a transporter, and it turns out that it's facilitating movement via a secondary active transport. And so how it's facilitating this is through this symporter moving iron 2, but also facilitated by the movement of proton. All right, so we have protons that are just facilitating the iron being brought in. So you're using a proton concentration gradient and the electrical potential of the membrane to drive iron 2 uptake. Okay, so that's how it's being coupled. All right, so here we get a cartoon representation of what's happening. Okay, so as I mentioned to you, iron 3 would have to get reduced to iron 2, or you might have iron in the form of iron 2 heme that is then going to be recognized by a receptor, and that's going to bring it into the cell, and then you can get access to that iron 2. So remember how I was saying that it's already in this oxidation state, but it's a matter of how do you make it accessible. So over here, that iron 2 is getting transported via the divalent metal transporter through the movement of protons. Okay, so now you have your iron here. Okay, this is now inside the intestinal cells. And so you're going to have storage of some of that iron, okay? But you're also going to have now movement of the iron from the intestinal cells into the blood. And here is a very critical event. The critical event is a change in oxidation state from iron 2 to iron 3. And so let's talk a little bit about this process. Also, the iron 2 is exported across the enterocyte membrane into the bloodstream by by a protein called ferroportin, okay, IREG1. So this is a transporter to move the iron 2. There is a critical oxidation event that takes place, release the iron into the bloodstream. So now we're converting iron 2 to iron 3. And it's this enzyme, the hepcidin enzyme, which is a copper-dependent ferroxidase. Ferroxidase means something that will oxidize the iron, and it's a copper-dependent protein. A related protein, which is copper-dependent ceruloplasmin, also plays a big role in terms of the regulation of the iron oxidation state. And you will see that copper and iron are quite interdependent in terms of being facilitating their bioavailability. All right, so we're here, this process of iron becoming iron 3. Well, then that iron 3 that's going to be delivered into the blood, well, there's a really important protein called transferrin that will uptake that iron and then will transport it to the rest of our cells. Now, why is that important? Well, this is an important event because of the solubility of the metal. So let's talk a little bit about the solubility of iron 3 with respect to the solubility of iron 2. Well, it turns out that iron 2 is extremely soluble, and I will mention to you why it's important that we need to oxidize it. So in the absorption from the intestine into the blood, that leads to iron becoming iron 3. But iron 3 is quite insoluble. At this pH, there will be a precipitation event. So this precipitation event, I'm just giving you a representation of how the iron could be precipitated, so iron hydroxide. And so if we wanted to quantify, well, of that amount of iron that gets precipitated, what remains soluble? Let's see how much relative iron would remain soluble. So let's focus on iron hydroxide, in this case trihydroxide, as our representation of precipitation of iron 3. If we were to focus on its solubility product, remember solubility product is a measure of the dissociation of the iron, in this context, from the hydroxide anions. Well, it turns out that the KSP, the solubility product, is 10 to the negative 38 molar. So from this information, we can derive a relative concentration of iron. And how is that? Well, if we're focusing on a pH of 7.4, right, pH equals to the negative log of the concentration of proton. Well, from this information, we can determine how much hydroxide is present, and from there we can determine how much iron is present. So you just have to do a little bit of math manipulation to determine that at here pH of 7.4, doesn't really matter, pH 7, a concentration of soluble iron would be 10 to the negative 17 molar. That is a very, very low solubility. Okay, that essentially that wouldn't be too biologically available. Okay, it turns out that iron is much, much more soluble than that concentration. But before we answer how it is much more soluble, let's answer this other question. But why even oxidize iron 2 when it is much more soluble than iron 3? Wouldn't it be more logical to just keep iron 2 so that it can be actually useful? Well, it turns out that the body carefully regulates the oxidation state because iron 2, although it's quite soluble, it also is quite toxic. And that is because iron 2 is very redox active, that it can generate reactive oxygen species. I'm not trying to say that iron 3 isn't, but iron 2 is much more capable. And so it can generate reactive oxygen species, or ROS, via Fenton chemistry. And if uncontrolled, these species can cause great harm to the body, harmful cellular damage when there is a loss of ROS homeostasis. And so this is something that the body has developed its mechanism for controlling. So iron in this oxidation 3+ has to be regulated completely at the level of uptake. What do I mean by that? Well, as soon as the iron 3 is formed and is going to get released to blood, you have to then grab it because, right, if you don't grab it, as I already mentioned, it will precipitate. Okay, so then it becomes unavailable. Or if you don't grab it, it will get reduced back to iron 2, and then it'll be extremely toxic. So what we have then is a really important protein, transferrin, that will bind that iron 3 and will facilitate its movement through the blood to our cells. So it will supplement the iron that we need. There is some amount of role of small molecules in terms of facilitating movement of iron, but the predominant form of iron in a healthy human being will be iron bound to transferrin in our blood as far as the transport. Now, that is not to rule out iron that exists in our red blood cells in the form of hemoglobin, right? That's iron that's available to move oxygen, but this is iron that's in our soluble blood to be able to be transported and used. All right, so let's talk about then transferrin and how it's enabling this movement of iron so that it's transported. All right, so we already talked about the mobilization process. Now we're talking about the transport process. Well, transferrins are a family of proteins, and they can exist in a monolobal form or a bilobal form. The dominant form today is the bilobal form in most organisms. I mentioned transferrin in our last lecture because we looked at mass spectrometry of iron bound to transferrin. Now we're going to look at more at its biochemistry and its coordination chemistry. Well, in its bilobal form is 80 kilodalton, and that is the form that exists in our bodies. And this protein plays important functions. So, as I already mentioned, iron transport, but also facilitates the homeostatic control of iron. In addition, it plays a role in terms of bacteriostasis. And so again, what that means is that you want to control the levels of bacteria. And so it's not that we want to, deprive our body of any bacteria. If you guys are familiar with the concept of the microbiome, we actually have bacteria that we depend on to survive. But we want to control the accessibility of iron. And so transferrin allows for this, in addition to another protein, lactoferrin, although lactoferrin probably plays a more direct role with pathogenic bacteria. So transferrin, if you want to think about it as a metal chelator, it is a very strong iron chelator. And so on the left side, we're looking at the coordination that is facilitated by transferrin. In terms of the binding site, we have 2 tyrosine amino acids that will coordinate to the iron. We have a histidine, and we have an aspartate. And if you notice, they're actually, in each of the lobes of this protein, there are 2 of these metal binding domains. In terms of their primary coordination sphere, in terms of how they coordinate to the iron, they are identical. So in addition to those 4 amino acids, there is this small molecule right here, carbonate. And that carbonate is referred to as a synergistic anion. The reason why it's referred to as a synergistic anion is because it is anionic and it helps the iron to be stably bound to the protein, so that carbonate is serving a role for stabilizing the metal and keeping it bound to the transferrin. So the net effect of the coordination coupled with carbonate helping it out is that you have very high affinity. And so this is the affinity of the iron to both lobes. At the C lobe, you have a constant of log K of 22.2, and at the N lobe, 21.3. All right, so let's remind ourselves about terminology, excuse me. Remember I said to you, when I refer to proteins as N or C, I'm referring to the two ends of the protein, the N terminus and the C terminus. So there's one lobe that's associated with the N terminus and another lobe that's associated with the C terminus. All right, let's characterize the coordination chemistry even more. So I talked about the amino acids that are bound to the metal, the carbonate. Well, then what other spectroscopic features can we focus on? Well, now we're invoking the stuff that we talked about, electronic spectroscopy. Well, it turns out that iron binding to the protein produces this really characteristic pink peachish color, okay? And that is due to a ligand to metal charge transfer absorbance at 470 nanometers. So that is being facilitated by the tyrosine amino acids, the excitation of electrons from the tyrosines to the iron, and you get this characteristic color. And so here you have the spectrum of iron-bound transferrin, which is typically referred to as hollow transferrin, versus apotransferrin, apo referring to metal-free transferrin. And so if we were to look at this in terms of extinction coefficient, well, this is a ligand metal charge transfer, so we should expect a relatively high extinction coefficient. And there are two ways that you can measure this number. This number can be in terms of protein concentration, so it's about 5,000 per molar per centimeter. Or if you measure this in terms of iron, remember there's 2 irons per every protein, well then this number is 2,500 per molar per centimeter. Okay, it's just the way you want to represent this number. All right, so characterizing then the metal binding affinity, well, other people have explored other metals binding to this protein just to get a general sense of the selectivity of this protein for iron, because again, this is an iron transporter. However, if you look at the what's coordinating to the metal, this obviously has the ability to bind other metals, and it's very good at binding other metals. And so people have characterized, well, how effective is it at binding certain types of metals? So what they did was to explore the Lewis basicity of the protein. If you think about the protein as a chelator, it's quite Lewis basic. And so that is what is established here. Compare the affinity of metals to the protein versus their affinity to hydroxide. Remember, hydroxide would be a hard base. And it turns out that you see this correlation is actually impressive in terms of its linearity, which means that what we're establishing here is that the serum transferrin as a chelator is a hard metal binder. In other words, transferrin is a hard Lewis base. All right, so it's going to have a higher affinity for harder metal ions. And so iron, according to this relationship, okay, is right here. So it's a higher end. But as you know, this protein is selective for iron, or generally selective for iron, right? So there are other things that are going to be involved in terms of that. All right, so when iron binds to the protein, what you're going to see is the effect on the structure of the protein. So here you have apo protein, and then over here you have the crystal structure of iron-bound protein. And so what we're looking at is what happens to the percentage of, what happens to the conformation of the protein. And so we have here the representation of the C lobe, and here we have a representation of the N lobe. Okay, so what happens is when the iron binds, you get a closure of the two lobes, sort of like a Pac-Man representation. This lobe will close, and this lobe will close. And we see that as depicted by the structures where the iron is bound to the protein. Okay, so we see a change on a tertiary level. So, excuse me. As a consequence of this change, sorry, I'm having difficulty here. Okay, as a consequence of this change, you have iron that's stabilizing the two lobes, and we will talk about that, how we can perceive that stabilization. All right, so focusing on the metal binding residue, I'm only going to look at the end side for a moment. So what we're going to see is on the end lobe, we have the 1/2, the N1 side of the lobe, and the N2 side. Where in the N1 side, we have these two amino acids, the aspartate 63 and the histidine 249. So I see that you have a problem, but I'm actually recording this so you guys can see this later on, because I can't fix something that's a little bit out of my control. All right, so aspartate 63 and histidine 249. So here, what's going to happen is you see that you have the presence of these two amino acids here. And then on the N2 side, you have the presence of the two tyrosines. So what's going to happen is that you're going to have the iron coming in, and that's going to lead to then a change in terms of how close those amino acids are. So here's a representation of the C site, where you have the corresponding amino acids on the two sides of this actual domain. So when it comes in, you have a closure. And now, in addition to just the coordination of the proteins, of the protein amino acids, you also have the presence of the synergistic anion in both of these sites. Now, this is going to be a really important to look at then. So we have the primary coordination sphere of the amino acids coordinating to the metal, the synergistic anion. Then you have additional factors that are going to be quite favorable in terms of stabilizing the coordination of the iron, where you have the carbonate interacting with arginine, and over here as well, that's going to basically stabilize the coordination of the carbonate. And if you stabilize the coordination of the carbonate, you're helping to lock in that iron. In addition, both the N lobe and the C lobe have other amino acids that help to stabilize that iron. And we're going to talk about how they facilitate this. So we look at the N lobe, okay, there is a process known as the dilysine trigger. So let's talk about what does that really mean. Well, it turns out that the coordination of iron to the protein at the N lobe results in two lysines becoming very close together and interacting. If you were to focus on the pKa of the lysine, the pKa is 10.53. Okay, and so the lysine should be positively charged at a pH of 7.4. Right, you would expect this. It turns out that when you have the ion coordinated, well, these two would come in contact, and there's actually a dissociation or deprotonation of one of the lysines, leading to a hydrogen bond between the two lysines. So the coordination of the iron that's nearby but is not actually interacting with the lysine results in a decrease in the pKa of the lysines, enabling this hydrogen bond to form. Okay, so the iron binding and the close conformation results in these two lysines coming in contact, and that enables the coordination of iron to be even more facilitated. And in fact, this hydrogen bond interaction is stable even at a pH of 5.5. All right, so what else is helping to facilitate this? Well, what else helps to facilitate the interaction between those lysines is a hydrogen bond interaction because of a hydrophobic box that is created by the tyrosines. Okay, so the two tyrosines and also the histidine that are available at the metal binding site. So the histidine, the tyrosine, and the other tyrosine creates this hydrophobicity that also enables a stabilization of that iron center. So if we were to review a little bit about hydrogen bonds, and we know that individual hydrogen bonds are not necessarily have a very high interaction in terms of energy, but there are, in distinguishing between a linear versus a bent hydrogen bond, there is a difference in terms of how strong that hydrogen bond is. Whereas if you have a linear interaction between the donor and acceptor of the hydrogen bond that actually ends up being stronger than if you have a bent orientation of that hydrogen bond. And here I give a perspective of the relative strengths, where if you have a donor acceptor distance of 2.2 to 2.5, you have a relatively strong bond strength that's almost at the level of a covalent interaction, even though it's not an actual covalent interaction. As you get further away, okay, then you're going to have that interaction be more electrostatic, so it's going to be weaker. And even further away, as you get out to about 4 angstroms, now this hydrogen bond interaction is even weaker. So you can see how hydrogen bonds, depending on their nature, can be relatively strong to weak, but then how you can facilitate a stabilization of this. All right, so when we're talking about the stabilization then, any sudden influx in protons, okay, would disrupt this interaction, and it can lead to what is known as a trigger of conformational change. That is the premise behind the title, the dilysine trigger. How do you go from a closed conformation into an open conformation? Well, that is because of that. If you have a sudden increase in pronation, the two lysines will become pronated, and then that two lysines becoming pronated will then lead to a possible metal release. Metal release. Now I will say to you, this was a traditional model that people thought that as soon as you have an open conformation, you will not have any metal. But crystallography has been able to expose a lot more insight into metal coordinations, particularly iron. And so this was a structure that was obtained in 2012 that shows to the two tyrosines. We do not have coordination of the aspartate or the histidine, but nonetheless, even though now you have this open conformation, the presence of the carbonate in addition to the sulfonate anion allows that iron to still remain bound. So even though we are in an open conformation, iron is still able to be bound. All right, so when I was mentioning to you then, this idea of trying to stabilize the interaction in terms of a hydrogen network, well, we also see this, in addition to the N site, we also see this in the C lobe. Okay, there is what is called a pH-sensitive triad. So you have the interaction between three important amino acids, lysine, arginine, and aspartate. And what I'm showing you here is the protein in different organisms and how we are able to see their interaction in the C site. Whereas in the human transferrin, that's HTF, we see the interaction of hydrogen bonds not as extensive as you see it in the pig version of the protein or as you see it in the rabbit form of the protein. Okay, but this is actually a really critical because you can see how this interaction is favorable in order to keep the metal bound to the protein. But then how acidification can potentially play a role. As it turns out, because of this extensive hydrogen bond network, this type of interaction is even stable at a pH of 5.5. And so I'll talk to you later about why this is important. All right, everyone. So in our next lecture, we're going to continue on in our discussion about how iron is coordinated with protein and facilitated transport into the cellular environment. All right, so as you know, I am recording these lectures, so I will post them up into the website. If there was any technical difficulties, you can review the recording. All right, guys, I'll see you next time.
Thank you. Have a nice
Professor, can I speak to you in private?
Sure, I don't know who's talking to me. Who's this?
John Steven.
Okay.